The rate and timing of cell division in your body normally are very precisely regulated. Cells are formed, mature, and eventually die. As this happens, new cells divide, creating replacement cells. Chemical messengers that pass between neighboring cells help keep the rate of cell division equal to the rate of cell death. Sometimes a cell breaks free from its normal restraints and begins to follow its own pattern of cell division. This precancerous cell divides more often than normal, eventually producing a mass of cells that also divide more often. Further changes in these cells can increase the frequency of cell division even more until eventually a cancerous tumor develops. At this point, the tumor grows large but is confined to the tissue where it originated. Late in the development of cancer, some cells may gain the ability to move into blood vessels and travel to other parts of the body. Once established in new locations, these cells continue to divide in an uncontrolled fashion, eventually producing new tumors. Normal cell division in the body depends on a precisely regulated set of events that determine when a cell will divide and when it will not divide. Two types of genes called proto-oncogenes and tumor suppressor genes are primarily responsible for this regulation. When mutated, however, proto-oncogenes can become what scientists call oncogenes, genes that stimulate excessive division. This situation is similar to getting a car's accelerator stuck in the downward position. A cell that experiences such mutations tends to divide more frequently than it normally would. In contrast, mutated tumor suppressor genes can become inactive. A cell that experiences a mutation in a tumor suppressor gene loses some of its crucial breaking power. Again, the result is a tendency for the cell to divide more frequently than it normally would. For a cancerous tumor to develop, mutations must occur in several of a cell's division-controlling genes. These mutations disturb the balance that normally exists between signals that stimulate cell division and signals that inhibit cell division. The result is uncontrolled division.